I'm going to do some stuff similar to Molly. I'm going to talk about our yeast synthetic population resource. And there'll be a brief point of the talk where I think there's some slides in common, but I think we can go over them again quickly, and then it will diverge from her talk again. So the model of adaptation that we all kind of learned about as undergrads is the standard model, where we have a mutation that either occurs or is pre-existing. We have a change in the environment, and then that allele or variant has a selection coefficient associated with it, S, and then the dynamics of that allele or the change in allele frequency with respect to time. Once the allele frequency is bigger than 1 over 2n, and the selection coefficient's bigger than 1 over 2n is roughly deterministic, and we expect to see that allele, as long as S stays constant, go to fixation. And so this is the standard Fisher model we all learned about. And what's important here is that although you, again, as undergrads, you were exposed to a lot of complex equations for this, if S is small, the change in P is roughly proportional to 2pqs. So in fact, the equation that predicts the change in allele frequency is very simple. And so that also explains why for rare allele frequencies, and as it approaches fixation, it slows down, because as you get close to 1 or 0, that the product of PQ becomes quite small, right? And so so basically under the standard model, a new mutation has to drift up to a high enough frequency that selection can see it, and then it actually sits around close to fixation until drift gets it the rest of the way. Right, so that's one of the dirty little secrets of population genetics and finite population sizes. Of course, what's interesting is that although that's the underlying genetics model that we all think about and learn about, that's not the evolutionary model we talk about or learned about. How many people? So I used this slide in a talk to a bunch of systems biologists, so no one could identify the common name of the species. How many people know the common name of the species? Really? Enrique, help me out. Will you? Yeah, these are Darwin's finches, right? So one of the things that led to that was really important in Darwin's synthesis of his ideas was that when he was on the Galapagos Islands, he noticed these finches, despite being very closely related bird species, had extremely different beak sizes, right? And there's a set of classic experiments that are 20 or 30 years old now by the Grants, where they actually studied these birds in the field. And what they saw was that when El Nino events happened, that the environment changed in the Galapagos. They could actually measure the selection directly on these beaks and showed the beak sizes changed quite rapidly. It's pretty clear that these beaks why they were changing was that following an El Nino event where the actual composition of seeds on the island changed such that the birds had to specialize in harder or softer seeds, and the beaks are an adaptation to that. But anyway, that's pretty clear from the work of the Grants and, of course, Darwin's initial observations that when we talk about evolution, we probably think the variation in these beak sizes, and there's a lot of evidence of that, is due to several genes. And the other part of the model that's sort of a little secret is that we kind of tend to think that maybe these characters, these quantitative traits, are sitting fairly close to a phenotypic optimum. And then there's some environmental change, like think about global change or something, which changes the optimum for that trait. And so what happens is when the environment changes, the most fit phenotype is different, and then that character will move to that new optimum, often very quickly, right? And so this idea is not controversial at all. In fact, Christian's lab proves that traits can adapt extremely rapidly to environmental shift. And we also know it from, I like this example where all these different breeds of dogs have been selected from a common ancestor that probably only existed a few thousand generations ago. In fact, quite many breeds are quite modern, in fact, right? So there's all sorts of standing genetic variation for phenotypic traits, behaviors, you name it, that you can select on extremely easily. And most of that change is probably due to standing variation, not new mutations. We also know that evolution can happen extremely fast, right? So I think a lot of these population genetics models, in fact, if we go back to this little toy example, the time to fixation of this allele, and no one ever looks at the x-axis, is like 7,000 generations, right? But if we actually think of some of these classic examples of evolution, we could think of the moths that Enrique talked about, blind cavefish, the evolution of maize from teosinte. These sorts of bursts of phenotypic adaptation are happening over hundreds of generations in many cases, not thousands, or maybe 1,000, okay? So what's that? So I think what's interesting is there's actually been some modeling of what's happening genetically under the hood in terms of these, what I call quantitative genetics models of adaptation. So again, the old dogma that goes back all the way to the modern synthesis is that phenotypes can quickly move to a new optimum in tens of generations, and that change is due to several loci. And more recently, a few groups have started to model what's going on under the hood, including Nick Barton, Guy Sellis, and I really like the work of my colleague Kevin Thornton. This was a preprint on bioRxiv, which I think is really worth doing in your journal club, but you might want to have two sessions to discuss it. It's a little thick. But here's basically when he simulated that, basically that Darwin-Finch model I talked about, where he basically had a population that was basically at a phenotypic where it was selected for a phenotypic optimum for a long time, such that it was allowed to reach an equilibrium where new mutations coming into the population were being balanced by their removal by natural selection. And then right at this green line here, he basically shifts the optimum some distance. And then what you see is that the trait, as we see in all the experiments we do, quickly moves over 20 or maybe 100 generations to this new, such that the phenotype moves to this new optimum, right? And then it stays at that optimum. And that's what we see in many of these experiments, like Michael Rose's experiments that Molly talked about, where she could move these lines to the new optimum very quickly, and then they don't really change much more because that's what happens. But what's happening to the genes under the hood under this model, right? Well, what's happening is actually very interesting if you start to simulate it. First, and there's a few things here, right? Is that what's happening is that you have these, what Kevin saw was that you have these genes or these alleles that are generally very rare in the base population that are driven up in frequency following the peak shift. Here's one here, here's another here. And then once the population gets close to that kind of new optimum phenotypically, things get really interesting. So you might think, right, your intuition might be that once it gets to the new optimum, all these alleles are now neutral and will just drift. But in fact, that's not what happens at all. In fact, what happens is that these alleles continue to change in frequency, and if you do some work, you can show that these changes are much more rapid than drift. So it looks like even once you reach the peak, there's still selection moving these genes to fixation, or sometimes they go up in frequency and they go right back out of the population, right? So that's the first thing, and I'll try to get your intuition up to why that's happening. The second thing he observed is that, again, almost all the adaptation is due to rare alleles of large effect. And the reason for that is if there is a rare allele of large effect, it's highly favored by natural selection under the peak shift. But alleles of large effect can only be rare because if they're rare, it means if they're of big effect, they have to be young, or natural selection will have removed them from the population. So when he actually runs the simulations and allows mutations of different sizes to occur, almost all the adaptation is due to large effect mutations whose origin is actually extremely close. And here's the origination times. It's extremely close to the time the peak shift occurs. So it's actually a fascinating and novel result, I think. So that's paper worth reading. Why do the dynamics happen that once we reach the peak, these genes are still changing in frequency? Well, it actually turns out that's exactly what's predicted. And it was written down in an equation by Alan Robertson, who's my academic great-grandfather, in the Journal of Genetics in, issue 54 in 1955. I meant to have the year there. So what happens is that when you get to that optimum, this is an approximate equation for the change in allele frequency. So it's proportional to the effect size of that mutant, which is A squared, times its heterozygosity, or like so much like that, the change in selection under the standard model, times the difference between Q and P, right? So what happens is this difference here determines the direction of change, and basically what's going to happen is if the allele frequency got over 50%, it's going to go the rest of the way to fixation, and if it didn't go to 50%, it's going to be lost. So that's really weird. And that 50% depends a little on what you assume about dominance, okay? So that's under an additive model, okay? So why does that make sense? Sorry, I'm not, might have to go manual. Okay, well, so here's what's sort of happening. That once you think of this population, if you think of two genes here who get close to this new optimum, an A locus and a B locus here, where the A has a bigger phenotypic effect such that those three genotypic classes are spread out more in phenotypic space along the x-axis. What's happening is that A locus is contributing a lot more to the variance in fitness because there's outliers, right? So the way to actually minimize the variance in fitness is to increase the frequency of the big A allele and then move the little b allele to compensate and hold your phenotype at the optimum, right? So what should happen under this model when you get to an optimum is that the load for the big A allele is minimized by actually driving it to fixation and compensating with this allele of smaller effect. And then once you get rid of A, you kind of deal with B and use a smaller effect allele still to kind of compensate, right? And so that's exactly what Alan Robertson's paper was about in 1955. So eventually, the way to minimize the variance in fitness is to fix the big A allele and then move the frequency of the B allele that has less variance in fitness to kind of hold you at that optimum, right? And so I think that's probably what's happening under Kevin's model, where even once you reach the new optimum, you still have deterministic changes in allele frequency, although you can see an allele keep going to fixation or an allele that was rapidly driven up in frequency being forced back out of the population, which argues for time series data. Yeah. Absolutely. So the idea, so this I've drawn, this is the loci contributing to the trait. And so the idea is that at the new optimum, the trait's under stabilizing selection again, such that things that are far away from the optimum have lower fitness, right? So this is a model where fitness is proportional to your deviation from the mean. Right? Yeah. 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 Yep. It's a second order thing, right? It should slow down. That's right. The mutation selection balance before the peak shift. Yeah, but Kevin's point is that even though you're at mutation selection balance, there's a bunch of mutations that happened yesterday of pretty large effect because they're always occurring. So if you just write the model down, you're allowing mutations to occur and they have their effect sizes are drawing from a gamma distribution. There'll be some whoppers there that happened very recently. There'll also be smaller effect alleles that happened much longer ago and have drifted to a slightly higher frequency. But natural selection doesn't care about those. It's going to take the rare frequency whopper most times. So it's kind of this fascinating result. Huh? Absolutely. But again, like if you have a model where where you're where if you have a large effect size, those rare homozygotes are far away from the population mean, so they have a lower fitness, right? So it's hard for them to get to a high it's hard for them to increase in frequency without changing the phenotypic mean or other things changing. So it's hard for alleles to come into the population and get to a hhigh frequency unless they're small effect. So it's, I mean, you know, and a lot of the human genetics models now have this assumption where frequency is proportional to effect size. I think I can't remember. I should know, but several, like maybe maybe a dozen genes on a chromosome. But then within a gene, you have multiple mutations occurring and being eliminated by natural selection. Well, that's a good question. But if you have 1,000, if their effect sizes have to be smaller, or they're invisible to natural selection, right? I mean, so that's the big debate in the field, right? Yeah. Yeah. No, this is totally additive within additive between loci. Yeah. In fitness, yeah, because it's kind of like a, yeah, yeah, yeah. That's right. Additive phenotypically epistatic with respect to fitness, because that's true with all these Fisher sphere models, right? Yeah. Which it's it's of that family, right? Yeah. Right. But Dimitri would argue that's the dominant mode of evolution. No, 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 no. Well, Kevin, of course, greatly simplified it such that you have a long time to reach an equilibrium, a single event. Yeah, it would be much more, yeah, yeah, absolutely. Yeah, yeah, yeah. But but I think Kevin's model's extremely, like, you know, all models are wrong. Some are just more useful than others. I think it's very useful because it's simple enough that you can understand what's happening, but without, yeah. Read the paper. You love it. I think it's really, yeah, yeah, yeah. Okay, so I guess one of the things I'm very interested in is trying to distinguish between these models, and I think using experimental evolution as opposed to some sort of comparative method is one way we can get at that, right? But of course, if we want to model what's happening under a model like Kevin's, that's a model that applies to sexual populations with standing variation, right? Which is what Darwin's finches are, which is what humans are, which is what a mouse is, right? So it's pretty clear now that if we want to model evolution in sexuals, we need standing variation, because that's what sexual outbreds have, and it's pretty clear that if you have standing variation, a lot of selection, at least initially, is on that variation. We want population sizes that are maybe approaching that of a species. That's because if we really want to give mutations a fair chance to matter, the total adaptation due to new mutations is limited by 2 times the population size times the mutation rate. So in a small population, mutations never have a chance to matter. And then we also need sexual recombination, or we're going to have evolution's going to be dominated by what happens in non-sexual microbes, where a mutation at one position in the genome is totally determining what happens to the entire genome, and mutations far away, and you get these clonal interference dynamics. And that's extremely well established in asexual microbes now, that this idea of clonal interference. I mean, it's kind of would be, you know, heresy to say it's not occurring. So again, if you have, in the absence of sex, if you start out with a whole bunch of haplotypes, what's going to happen is you're just going to have one haplotype quickly dominate the population, right, genome-wide. And so what sex is going to do is it's going to allow different hunks of the genome to evolve independently. Okay, so that's why we think we need a model that can do, to model these things, we can do that. So as Molly, this is data from an old paper of ours where we did a highly replicated experiment in E. coli and looked at where all the mutations are that many were probably important in adaptation. Although bacteria have big N, they're asexual, so adaptation is necessarily through new mutations, and the dynamics of those mutations is impacted by clonal interference. So great system, but we can't really model evolution in sexuals using it. And this is just this is from Greg Lang. The name looks similar enough to my last name that I could try to claim credit for it because I love this figure and I use it a lot. And the Scilabs makes brilliant figures. They must have a graphic artist on staff. But under clonal interference, of course, we're going to have new mutations arise. So this is time here along the x-axis. We're going to have new mutations arise and increase in frequency such that they can be at a fairly high frequency in the population. But those mutations are competing against other mutations that occurred and increased in frequency, such that you can have a mutation like this one that increased in frequency and then was lost. You have this mutation that was increasing in frequency and looked like it would have been outcompeted, except for it picked up a second set of mutations that on that haplotype that let it survive a little longer and eventually picked up additional mutations that let it eventually dominate the population. But the point is, in asexuals, clonal interference means that you're always competing against other mutations, and ultimately your fate is determined in many cases on if you pick up additional mutations on that background that help you get there. And that's what our earlier talk was about, I think, to some extent, right? Okay. What about Drosophila? I mean, there's a lot of advantages to Drosophila. They're sexual. They have standing variation. It's hard to keep N above 1,000. It's hard to do a lot of replicates. They have a fairly big genome size. You're also a little limited in that it's hard to do experiments lasting more than a few hundred generations. But you can certainly, they're a great system for applying an environmental shift, and look what happened. Of course, we've done some work in Drosophila, myself and Molly, and have done resequencing on it, and I think I won't talk much more about that. So basically, the classic systems are in some ways not optimal for addressing this question, although flies have sex and standing variation. They are limited in the number of generations per year. It's extremely hard to do big population size experiments, although we could do them. Time points can't be frozen and revived. Gene replacement's still difficult, although there was this paper that came out a month ago, the prime editing paper, that I think is pretty exciting. In bacteria, again, they have all these features except for the no sex and no standing variation. So Molly and I created this system we call sexual outbred yeast. So we basically create an outbred base population by crossing natural isolates of yeast. So once we have this system, there's the potential for lots of replication. We think we can maintain fairly or pretty large population sizes, depends on the experimental details. We can force recombination. We can potentially carry these experiments out for thousands of generations, although you'll see the life cycle isn't strictly sexual, right? So it's important to note that there's only a few people doing this. Molly, myself, Gianni, the Scilab has a project doing it, although most of their work isn't in this system. So this differs from classic yeast experimental evolution, where the evolution starts from an isogenic base and is strictly asexual, which is the dominant yeast paradigm, which in many ways genetically is very similar to what's going on in bacteria, right? So we've tried to change this. What's important to note that this is a completely synthetic or artificial system, right? Yeast are if you collect yeast in nature, they're almost always completely inbred and isogenic. They have sex extremely rarely in nature, as far as we can tell. Okay, although they do sporulate in nature. When they do sporulate, they tend to mate with an ASCII mate, with a full sib itself. What Molly talked a little about our first kind of foray into this system, where we used a 4-way cross that Gianni had done. What we've done since is we've just sampled more extensively from the tree of this system, and we've done an 18-way cross where we've brought in where these are all the strains we're looking at. They're basically a worldwide sample. There's a few you can see, like B10 and A10 are missing. These were basically Malaysian strains that we couldn't mate very well with the other strains. So it's important that these things are all crossable to one another and sporulate well. The reason they're designated with an A or a B is these A populations were all brought in as a mat alpha, and these B populations were all brought in as a mat A. You can see B1 through B4 are the same as A1 through A4, except for we've switched the mating type locus. So in yeast, you can actually, there's a single locus that determines if you're an A or an alpha, or a boy or a girl, if you want to think about it that way. It's not really right, but and we can just flip that locus and make you a true breeding male or a true breeding female. Okay, so that's how we do this cross. These first 4 populations are important enough and well characterized enough that we thought it was good to have them doubly represented in our base population. Okay, so that's what we're doing. So unlike Molly's experiment, we're starting out with 18 founders. For some reason, I have this figure here. I don't remember why. This is the yeast life cycle. So again, what we did was we take these alpha strains, we mate them to make a diploid A alpha. We're going to carry that through some sort of selection, and then we're going to occasionally sporulate it to get back to our haploid cells, and then repeat the whole thing. Again, this is all done by making permanent switches to the mat locus. One thing that's a little different in this set of lines compared to the ones Molly talked about is that in Molly's experiment and Gianni's line, we force diploidy using auxotrophic mutations on link to the mat locus. In these strains, we've put just a few kb downstream of the mat locus. We've put like a selectable marker, dominant selectable antibiotic type marker that's in LD with the mat locus. So the mat A has one marker and B has the other, and then we can plate these or put them in media with 2 selective agents. The reason for that is there's less load. It turns out by doing that, there's a lot less load involved in selecting for diploidy. So in Gianni's line, about half the lines half the diploids are lethal anyway, I won't go into that. So again, the cross is much like the one Molly described. We're going to take all the 12 mat A's and cross them to 12 mat A's, cross them to 12 mat alphas to make diploids, resporulate, and then we just carry that through meiosis and mating for 12 generations, which creates this highly recombined base population. Okay? Yeah. There are no genetic compatibilities between the strains. No. They're all crossable. They probably vary quite a bit in their fitness. So you'll see the base population is not, the frequency of each founder is not 1/18. In fact, it's quite variable. But it that creates the incompatibilities. Yeah, no doubt the incompatibilities are due to all sorts of reasons. these F1 diploids are viable, and they produce spores. So what's going to happen is that when we go through this process, those combinations are going to be eliminated. But then it looks like there's just some bad strains that just have poor combining ability that drop out genome-wide to a large extent. We also saw that in the fly synthetic population resource. So there's ways to do it to get more equal representation, but they're a boatload more work, as Molly's trying them, actually. Yeah, that involve doing the tetrad dissections at every step. Yeah. Which once you scale up to 18 founders is hard to get by. Okay, so this is why I showed this slide. So again, what's key here in these protocols is that we have these outbred genotypes, and there's this sporulation step. And if we think about what's happening, we're going to put these yeast on Friday in a what we call a sporulation media, which is basically a media without a sugar source. Of the diploid individuals that are put into this, it looks like fairly consistently over 70% of the diploids result in either 4 or 2 spore ASCII, and about 30% are actually decide not to sporulate. And then on Monday, we apply an aggressive ascus disruption and several nasty chemicals, which in our base population basically kills these vegetative cells such that they're only 1% in the population, and these single spores are 99%, and then we'll remate those, right? But why I show this is that what you're going to see a little later in the talk is that one thing that can happen in these experiments is that the yeast figure out a way to kind of get through this, and then we get the evolution of asexuality, okay? So I'll come back to how that happens, but it's important I hate to show it, but the life cycle of yeast sort of matters, right, for what we're going to see. And I'll come back to that figure yet again. So if we just kind of pick single clones from the base population and sequence them and infer haplotypes, so that, again, in this talk, I'm going to color the founder haplotypes different colors. In some cases, we can infer them in gray. In other cases, there are 2 founder haplotypes that aren't resolvable that get combined, okay? So we can see, though, that this is a single clone here. We can see, obviously a lot of changes between haplotypes. And again, this is a single chromosome. I'm just zooming, or an arbitrary chromosome I'm going to zoom in on so we can see the haplotype structure a little more. We tend to have these blocks that are, anywhere from 20 to a couple of 100 kb in size that define the base population. So in yeast, 100 kb might be 50 genes or 30 genes or something, right? So again, that we haven't, totally broken it up, but we've broken it up quite a bit. There's some evidence from Gianni's early work that additional rounds of recombination don't break these up much more, in the sense that these might be, there might be some recombinational hotspots that are limiting us. But that's the kind of size of the haplotype box in our base population. So again, if we look at I don't know if I have many of these plots, but these are the founders here. This is their frequency in the base population. The different colors here now are different chromosomes. So you can see like this AB3 or this particular haplotype did very well in making the founding population, such that it's often at a frequency of 20, 30% in many of the replicates. Some other ones did well, and then some are kind of poor, right? They're in there, but at a fairly low frequency, right? So in fact, here's the average frequency, such that some of the founders, their average frequency in the base population is less than 1%, although there's certainly hunks of the genome where they're at a much higher frequency, okay? So they seem to, we seem to get all the variation in, although certainly it doesn't all start at 1/18, right? Okay, and then Molly showed you this, and I think I'm close to diverging from her in a second here. The protocol's pretty simple. On Friday night, we basically put these the yeast in a sporulation media where they basically recombine and generate 4 spores. To a population geneticist or a yeast geneticist, we call this sex, right? Because that's where recombination occurs to make gametes. And then we isolate single spores on Monday, and then we combine those single spores to make diploids. So that's what yeast geneticists call the schmooze event, when the 2 haploids form to make the diploids, which is more what maybe people might associate with sex, not what the population geneticists do, right? Then we're going to recover these selecting for diploid using these dominant antibiotic markers, and then we're going to passage these things in 24-well plates. Unlike Molly's experiment, we're going to add different chemicals to the plates to create different selection regimes, and then look at what happens, right? So we're in the midst of a huge experiment where we're evolving the yeast in the presence of, I think, 16 different stressors. So we have, and these are the stressors. So we have like ethanol, DMSO, cisplats, frontline chemotherapy drug, caffeine, salt, nicotine. These are kind of pretty familiar drugs that basically we know we can dissolve in the YPD media and have been studied by other people. For each selection regime, we have 16 replicate populations we're going to carry through that, and then we basically managed to carry this whole experiment through 24 weeks, which is kind of hard, actually, because there's 384 total samples we're carrying. This data we're still analyzing, so I've decided to just talk about fluconazole today, because I had to arbitrarily pick one treatment, and even then a small number of replicates to try to talk to you about. It's a commonly used antifungal drug, right? So if someone is in a hospital and has a fungal affection, so usually an immunocompromised patient, this is one of the drugs they could be treated with. Yeah. Millions. Yeah, so the bottleneck is millions, but once you disrupt it to get the spores, there's still millions of spores, but it's not exactly clear, like when you schmooze them. So what's going to happen is they're going to schmooze, and then it seems to take, like the schmoozing seems to take a long time. And so some of the cells are going to come out of the schmooze quicker and start budding, and other ones aren't. So there's probably like a selective bottleneck there where if you go to being a diploid, some people can launch quicker than others, right? So the number of spores is quite large. Yeah. Of the what? Of the what? Yeah. No. So the evolution is in 24-well plates with like I think we found a mill, because of the shaking, it would come up the sides a little bit. So I think we're at 700 microliters or something. No, and then the mating and all that's in the deep well, 96-well plates, the big that hold like over a mill. So yeah, we've kept the N as big as we could, although there's the census N is not totally going to tell you everything, right? Yeah. I don't know, but I mean, if you count spores, yeah. Well, the diploid stage, I mean, once you have if you count the number of diploids coming out of the schmoozing, it's more than 1 million, always. The bottleneck has to be more than 1 million, but those diploids might be launching quicker, such that there's more variation, and they don't all have equal fitness, right? So the population size might be quite a bit smaller than what your census size. Yeah, but over the whole experiment, we never go below 1 million census. And then if once you let them grow in the YPD, their population sizes spike up to billions if you just look at cell density. Yeah, so big populations. But I think, yeah. But, that's all. Yeah, there's an asterisk. So we actually, because this was hard to do, this is going to work, yeah. We actually built a custom robot just for doing this experiment. So you can see, this is like, if I'd lived 400 years ago, I would have been building those automatons, right? And what we have here is so there's an array of 24 things that can pick up tips, filter tips. These are our 24-well plates. So that's the whole experiment there, 16 24-well plates. Each well is a different population with potentially a different drug. We actually punch right through the membrane. We never pull it off, and this is going to be a transfer from a 24-well plate to a deep well 24-well plate, where we're probably moving it to this because this is what we're going to sporulate in, for example. And I should be able to have it run here. My Lord. Oh, there we go. Yeah, there we go. So that's kind of, this going obviously doesn't go this quick in real life. There's actually a soundtrack, but it doesn't come out into the room. But it's kind of like, the soundtrack's pretty good. Not Justin Bieber, but pretty good. Huh? What? Well, so it actually took over a year to build. I had a bunch of engineers working with me. It still breaks from time to time. So but it actually, what's nice about it is unlike a commercial robot, it's doing 24 samples at a time, so it makes it quite quick. And the deck is huge. So we can lay out the whole experiment, and I think it leads to less problems with getting plates mixed up, because you can lay out the whole, we can have undergrads lay out the whole experiment, and then everything's barcoded and stuff, and it gets checked, or color-coded anyway. Also, it doesn't show it here, but the whole thing is enclosed in a laminar flow hood that we built. So it's kind of so we don't get stuff falling into the well. So it's actually was a, yeah, I mean, I could share the plans, but it would take a while to get up and running. You have to want it, really want it. Yeah, so I'll say. This slide's for Christian, of course. Contamination, so it's for sure happening, right? When you do these big experiments. So what's the contamination rate? I don't want to put a number on it, but I will say a few things. Yeast can't fly. Yeast are way bigger than bacteria. This is actually a picture showing yeast cells, bacteria cells. So these gas-permeable membranes, when we've tried to use them with bacteria, the bacteria just get through them somehow and into neighboring wells. They seem to be pretty effective with yeast. Like if you leave blank wells next to a well with something in it, they don't become contaminated that much. The 384 populations are not barcoded, right? Now, the reason for that is that we don't start from an isogenic strain. So if we tried to introduce a barcode into a population, we would probably really knock down diversity. So we haven't been able to really do that, okay? But that would be one thing we could do. One thing we do is the way those 24-well plates, and sometimes we go to 96-well plates, the way the treatments are laid out, the same treatments are never beside in adjacent wells in either 24 or 96-well plates. So if there is cross-contamination, it'll tend to be between treatments, and they probably don't establish that well. We carry several YPD no yeast controls. So these are just empty wells with the media yeast like to grow in. And so if a single yeast gets into a YPD well, it'll turn cloudy, and you'll know you had an event. So you can use those to kind of measure the rate at which they occur. we tend to see about once every couple of weeks or once a month, one cell lighting up. So that's about the rate it's occurring. Those YPD controls also serve as plate spin controls, which are very important when you're working with plates, right? Because plates can be put on a deck like this or like that, right? And if that happens, it's a disaster. Okay, so you need plate spin controls. When we actually start to sequence these, the genotypes look similar between treatments, not between neighbors on plate neighbors. So it suggests there's either not that much contamination or natural selections. Pretty efficient. And I would argue sample mix-ups are probably a bigger problem. So it's really important you don't mix up plates or go into a plate at the wrong position to pull a sample out. Okay, so that's that. So again, what I'm going to show you here for basically the rest of the talk is just 4 of the fluconazole replicates, just for giggles. Again, this is data that's less than a month old. So this is SNP differentiation. You can see quite a bit. So the different colors are different chromosomes, and the y-axis is change in frequency. You can see some of these things changing close to 100% in frequency, which makes sense because unlike Molly's experiment, things can start out pretty rare in this experiment, right? Because it's an 18-way cross. There looks to be some peaks that are maybe showing convergent patterns between the replicates, like maybe this and this, although it's a little hard to see what's going on from the single replicates. So that's the SNPs, okay? So because we start out with 18 founders and we know the founder, those founder haplotypes are known, we like to do the analysis at the level of haplotypes, not SNPs, right? Because what's happening in this population, if you think of this, you imagine there's a SNP here that's advantageous. That SNP is buried in either this haplotype or a couple of haplotypes. And so what's going to happen is if this SNP is actually being selected to increase in frequency, it's necessarily going to pull this purple haplotype along that's embedded in. And it's actually much, much more powerful to actually detect the changes in haplotypes than the changes in SNPs for technical reasons. So we prefer to study haplotypes, right? So now we're going to do a little Californian exercise, right? I want you to repeat after me because it'll make you feel good, right? SNPs are just markers. Haplotypes change in frequency, okay? It's really important because in these experiments, SNPs are markers, right? Because the amount of LD is such that we can rarely look at a single SNP, unless you're in kind of Robert's world where you have a couple of hundred natural isolates such that the LD is broken down in the first place, right? But when you're starting with a few dozen haplotypes, you necessarily have these big blocks of SNPs that are all showing the same pattern ripping around, right? So the selection's at the level of haplotypes, really, okay? And so it's often better to think about it that way. Okay, so again, let's get back. So now I'm going to show you some hard figures, which I'll try to walk you through. So I'm going to go back to this figure for a second. So imagine that there was let me see, I got to look ahead to see this. I don't know what that is. I'll get to there in a second. So yeah, again, what's going to happen is if I'm going to do this analysis at the level of haplotypes. And so what's going to happen is I'm going to take my pooled population sequencing, and at any given position, so say some little position of 50 kb or something, I'm going to try to estimate the frequency of each of these 18 founder kind of haplotypes, right? So I could say if I was looking at this position here at around centered on 400, I could say this light blue haplotype has a frequency of 1 out of the 12 or so I'm showing, and the orange haplotype has a frequency of 3, right, at this position, right, or whatever, and the yellow 2. So if I could actually take the pooled data and estimate the frequencies of these haplotypes, I know I can maybe estimate those quite accurately. And then once I know the frequency of the haplotypes, I by definition know the frequency of all these SNPs, because I know how the SNPs sit on the haplotypes, right? Okay, now what's going to happen here? Okay. Okay, so how do we estimate the frequencies of the founders? So there's several methods out there, including the people know the Novembre paper. We prefer to do it using a linear modeling approach, because I think there's lots of weirdness that can happen with real data that you have to be careful of. But it's pretty simple, actually. So if you imagine you have pool-seq data, and we're looking at a string of SNPs in the genome for some window, you have the frequency of that, of say the reference allele in the pool, right, for a bunch of SNPs. So the darker colors would be closer to the reference allele being 1, and the lighter colors, the reference allele being 0. And then for each of our founders, their sequence, so we know basically the haplotype in this window of the founders, and they're black and white because they're basically fixed for either the reference allele or the alt allele. And so basically this is just a linear optimization problem where we're going to take the difference between the frequency of this vector of SNPs in the pool minus the frequency of each haplotype in the founders times a set of weights, where the weights are basically the frequency of those founder haplotypes, right? Now, what's weird about this is if you just thought of these W's as betas, this is just linear regression, except for we have to constrain these weights to all be between 0 and 1 and to basically sum to 1 because they're frequencies, right? And so it's actually a constrained minimization problem, which it turns out there's a whole branch of mathematics for dealing with. There's actually a R language called limsolve, where you can do these constrained maximization problems. And people that do statistics for a living just know about this, and I struggled for several months before I figured it out on my own, and I had some sort of conversation with my great colleague, Larry Muller, where I discovered this R module called limsolve that lets you optimize least squares with box constraints, and it was a revelation. And he said, dude, using Lagrange multipliers in a constrained linear optimization problem is textbook stuff. Note to self, talk to a statistician sooner, right? So there's a little lesson here that don't leave your pride at the door and go talk to like people that think about this stuff for a living. But this is a good package to know about. Okay. Okay, so now I'm going to show you frequencies of haplotypes, right? So this is a funky plot, right, where there's 4 colors, right? And what I'm showing here is on the x-axis is location in the genome. On the y-axis is the final frequency of the haplotype in the population, right? And the red here is just the frequency of the most common founder haplotype at that position, okay? Which could be founder 1, it could be founder 2, right? So if we go, let me see where I am here. Whoops, that's not good. If we go back, if I had happened to drive this entire haplotype to 1, although it's recombinant for this chromosome, then that would be a red line at 1, right? Because the final frequency of the blue is 1, the orange is 1, this orange is 1, this whatever pink is 1, this is 1, right? So that's what I'm doing. That plot's a little weird, but right, because it doesn't tell you what haplotypes changed. I'm only showing you the most common haplotype, right? So what you see here is something pretty interesting, and we see this in all 4 replicates, that a single haplotype has increased in frequency in most places at the genome where there's been change, right? And the haplotype that's changed the second most, which would be purple or something, in most cases hasn't changed at all, right? And that's a very general result. And I'm very excited about this result, and here's why, right? So let's go back and think about models for a second, right? So imagine we have a SNP that's actually the causative allele, and that SNP happens to live on 2 different haplotypes, right? Well, what should our change in haplotype frequencies look like? Well, if this is true, and you have a well-isolated SNP that's at intermediate frequency on 2 different haplotype backgrounds, you should see the blue and the red haplotype increase in frequency, and the green and the purple go down, right? So the fact we're only seeing 1 haplotype going up for most cases where there's selection tells us that this model's sort of wrong, and we have to think of 2 different models. We have to think of a model where there's a rare variant that happens to be private to a single haplotype that's increasing in frequency, right? Or what's really happening is that there's several SNPs buried in a haplotype, and what's being selected for is the haplotype that just happens to have the most, like, who's the sum of the effects is like the biggest, right? Okay, so you either need this kind of, there's like several variants in a region that's creating a haplotype that's being ripped up, or there's a single rare allele of large effect, right? Okay, and so that's very general, seems to be a very general result, which seems... That's true. Maybe so, yeah. I don't know. Yeah, I think we have to more formally model it now. Yeah. But the point is that in these populations, it's very difficult. Like, I'm surprised I don't see any place in the genome where 2 haplotypes are moving up. We just don't see that. Yeah. Okay, so now, small divergence here. This is a painting by Jackson Pollock. Does anyone, do you guys know who he is? Who knows? Everyone knows who he is? Yeah, there's a good biography about him on Netflix. I don't know if it's on Netflix, but it was a good movie. It's quite old now. He was an American painter and very influential in modern art in mostly 1950s. I pulled this painting down. It's actually in the Museum of Modern Art here in Austria. They probably have several Jackson Pollocks, right? He was famous for, he could churn it out, man, right? Like, so he would come and there'd be a Jackson Pollock exhibit, and he would show up with no paintings, and he would just do them the day before, and then the paint was still wet when people were coming into the gallery, right? That was the famous story. So the reason I show you this is now I'm going to move to what we call Jackson Pollock, what we're calling Jackson Pollock plots. Molly had some of these, actually, but she didn't call them that. So now I'm doing what maybe is more natural, and you can see why I didn't do this initially. What I'm showing here is the most changed haplotype for these 4 replicates. Just the most changed, not the second most changed. And now I'm going to color the plots according to their founder haplotype, okay? So, and then we call them Jackson Pollock plots because they quickly become extremely busy and colorful, and in some cases, especially if you want to show 2 different, the most significant and the second most significant haplotype, they can look very busy. But you can see, of course, that what's being, so now you can start to see some interesting stuff when you look at the Jackson Pollock plots. You can perhaps see, like, here's the blue founder getting ripped up in at least a couple of the replicates. So you can start to see cases where you're not only seeing change in the same regions, but it's clear it's the same allele moving up, right? But this also shows you that the actual, what actual haplotype gets ripped up is somewhat random, right? Okay. So these are very hard to look at. So what I'm going to do is just zoom in on chromosome 7. So that's this chromosome, I think, or this one. I can't remember. I'm just going to zoom in on one chromosome to try to let you see what's going on. Yeah. What's that? 3. The blue one. Yeah, so it seems to pop up more than the others. Yeah. So here's that chromosome. So it's kind of neat. So again, this is at generation 12, I want to say, or week 12. And here are these 4 replicates. So it's kind of neat. You can see it almost looks to me like a modular thing where you can see, like, here's these 2 blue peaks getting ripped up here, right? This seems to be also showing up in this replicate, something, but not in these at all. Similarly, this thing here that's getting ripped up is perhaps appearing here, right? There's perhaps some overlap, but then it's not really showing up here. Right? So again, it looks almost like we're seeing, at least on this chromosome, evidence that there's multiple regions changing in frequency, but it's not all that replicable over the replicate populations, even though these things were starting out at frequencies of the alleles were starting out at frequencies of 1%, 1% or less, but the population sizes were much larger. So we should be in deterministic land. Initially, yeah, who knows? Yeah, that's physical distance, yeah. Oh, these are, well, I mean, again, if you look at those, if you go all the way back and look at those haplotypes, I mean, that's much, the blocks are much smaller. Yeah. I mean, obviously, I think the next thing would be to look at some clones, right? That what? No, this is after, at generation 12 for fluconazole. Oh, you're saying we shouldn't see that haplotype? Yeah, maybe it's. I mean, there's in the base population. I just showed you 12 haplotypes. There's probably a billion haplotypes. So that haplotype maybe exists in the base population. I don't know how common it is. Yeah, it's almost certainly a recombinant haplotype. And in fact, we'll see, I'll show you another slide where we can see that happening, I think. It could be that too. And in fact, in some cases it is. And I can show you that. I'll show you that. Yeah. So the other thing we can do is, of course, we have time points. So I can look at, yeah. I don't think so. So we've actually done complete de novo assemblies of the founders, and there aren't inversions in the founders, although they could have occurred during the evolution. So now you can see the Pollock plot starting to come to life here. What I'm showing you here is weeks 6 and 12 for those same two replicates. Since I've already basically used up my entire palette in R, you can't see the two populations here, but you can see something going on. And then I can, again, just zoom in on chromosome 7 for us to look at. So again, it kind of is what you think. That's generation 12, generation 6. So what do you see? Well, here we're seeing the claim from one of Christian's first ENR papers, right? Where we see this by generation 6, we have largely moved this haplotype to the frequency it's going to stay at, right? Although what's weird here is that by generation 6, we ripped this up here, and then by generation 12, we've also pulled this up. Over here, it looks like this is increased in frequency, although it started out as an entirely different haplotype running up, and then it went from red being favored to green being favored, maybe later in the experiment. Well, maybe, I don't know what that is. Yeah, that's regions where we can't, where that hunk of DNA is consistent with multiple founders. So that's where, like I said, that the haplotype thing has problems telling alleles apart, but you would guess it's green. Yeah. And again, you can see here in this replicate, we have these blue alleles that have reached a fairly high frequency by generation 12, but they hadn't really done much by generation 6. even though they were quite common to start with. So it becomes very hard to describe these dynamics, I guess what I'm getting at. But that's, again, what we're seeing. And it seems like we see, this for different, these examples aren't really cherry-picked. They're just 4 replicates, or in some cases 2 replicates, which I could show you other ones that look like this. So that's what we're struggling through with the analysis. So another thing we observed, if we go back to that initial plot, so this isn't a Pollock plot anymore. This is a plot of the red being the most common allele. That legend should be there. But the red is the frequency of the most common allele, and the purple is the frequency of the second most common allele. So these are, that's why that's there. So this is just two replicates here, not 2 I showed you. And then this is the Pollock plot for the same 2 replicates, right? So this is a weird pattern, right? That doesn't look like that earlier figure I showed you. What could be going on here that we have that the most common allele for most of the genome looks to be at a frequency of about 50%, and the second most common allele looks to be at a frequency of about 50%, yet those alleles are recombinant from the Pollock plot. What could we maybe infer from this happened? Well, there's a hint, the slide title. So what we think's happened is that a single diploid clone has figured out a way to get through our selection protocol and is ripped up in frequency. And so what we're seeing here is just some random kind of clone here, right? Some random recombinant who got through that selection regime and has come to dominate the population. So we believe this population is dominated by a single diploid individual that's escaped our sexual selection. So these plots are actually an incredibly strong signature for these events. And the reason we see the red up here closer to 1 is presumably that's a region of the recombinant genome that just happens to be homozygous for the same founder type, right? Okay, so we do see this evolution of asexuality, and it happens actually in like a large fraction of the replicates, right? So again, if we go back to this figure, presumably we think what's happened is in these replicates, we've picked up a mutation or standing variation that's allowed these diploid clones to get through our selection event, and then such that they're escaping this. And what we think's happening is that the actual when we schmooze the spores, it probably takes maybe 8 hours or something before that new diploid can start budding. But we believe that these escapers might be able to start budding right away and maybe get through like 2 or 3 cell divisions before the well-behaving things actually get through any. And so if you can sneak through this chemical disruption, there's a huge fitness advantage at the other end of the tunnel, right? So we think that's what's happening, and it's happening in quite a large fraction of the replicates. So luckily, again, we've replicated the experiment enough that we can identify these and just live without them. Is probably what we're going to do, or study them in their own right. Okay, so again, what we think's happening here is that there's a strategy that's evolved where there's basically a genotype that's not sporulating, then it doesn't sporulate and there's no food for 2 days, but it can somehow hang on. And then it also has some mutation that makes its cell wall incredibly strong or resistant to these chemicals, such that it can get through this chemical kill step on well, that would have been a Monday, not a Tuesday, but yeah. Okay, so we think that's a strategy that's evolving here that we didn't anticipate. Yeah. Huh? Oh, absolutely. Yeah, so there's probably some, so in some cases we're going to get partial ASCII disruption, and you're going to mate with a SIB, a full SIB. So there's definitely some inbreeding. And, it's this trade-off, right? Because if we disrupt the cells even more, we're worried we're killing the spores. I mean, yeah. Well, no, because the pattern we see is a single diploid genotype dominating the population. So there's like a consistent genotype that's just ripping up. Yeah, but if they're self in each generation, they should go homozygous. And we actually see that. We see some cases that I don't show you where we get a single homozygous, like a single recombinant thing getting to 100%, but I think that's a doubled haploid, which Greg Lang showed can happen in these experiments. Huh? Yeah, this is just generation 12, where they've been for several months, and then clearly we have this escaper that's come to dominate the population. Yeah, so we can look at these at generation 24, although it's a lot of work and we're not that interested, but yeah. Yeah, our guess is that it's more consistent with someone who's can get through that selection, but we're testing it directly. We can actually take these clones and see if they're able to get through this treatment. That's what we're doing. I mean, where there's like experiments that we can do. But again, what we see, the patterns of the data in the still early days looks like the decision to be sexual or not is a commitment. You have to commit to one or the other. You can't sit on the fence. So again, in about 50% of our replicates, these cheaters evolve. It depends a little bit on the selective regime. They don't always fix. The details are still being worked out. But in the cases where the cheating doesn't evolve, it's almost as if they double down on sex, and that the populations then seem stable, that they don't show cheaters later on, and the sporulation efficiencies in these non-cheating lines might actually be going up. We're quantifying that, so it looks like these events happen early, and then establish themselves, or they don't happen at all. We don't understand why. Again, it's something we're studying. Okay, what about aneuploidies? We can look at that. So again, if you're a yeast geneticist, a lot of yeast geneticists believe that all adaptation in yeast happens by aneuploidies. So if you take an isogenic yeast strain and expose it to some chemical, you'll often see the adaptation happening by aneuploidies. That's because they basically need to wait for a new mutation to evolve, and aneuploidies happen very frequently, right? And we see these in our line. So this is one of the treatments showing, one of the fluconazole treatments showing an aneuploidy for chromosome 2. And so this is basically all these individuals are 2N for chromosome 2, and there's basically a 3N chromosome 2 version that's sitting at a frequency of about 30% in the population, that's gotten to a frequency of 30%. Okay, yeah. Where? Here? Yeah, maybe that's like a small, maybe it's like a really low frequency. Yeah, could be. Yeah. Yeah, of course, the way these work is because it's we're getting it from pool-seq. So you're basically, what you're trying to do is say if the frequency of the chromosome different, is the coverage different from average for the rest of the genome, right? It's a little noisy. So you're way better off to detect these events when they get to a high frequency, right? Yeah, but this is an example of one that gets to we can estimate the frequency at least of the events that get, say, above 10%, where we start to feel confident that it's not an artifact. Yeah. No, well, that's the next slide. Yeah, so that's what I thought. I thought we were just going to see them in the cheaters, but here's in the fluconazole data set. I had Rob generate this table for me like the day before I left, where the green replicates, if I remember correctly, are the ones where the cheating didn't evolve. The white replicates are where cheating evolved. And these are all the aneuploidies that we observe that reach a kind of measurable frequency. So you can see a few things. One is that in all the fluconazole replicates, we have 1, 2, 3, 4, 5, 6 aneuploidies. They only reach a frequency of a max of 30%. So that can't, first of all, that's not the dominant way they adapt at all, but it's certainly happening. And second of all, it doesn't look like there's any relationship between those aneuploidies happening in like here it's happening in a cheater, a cheater, a cheater, a cheater, but here's a non-cheater and a non-cheater, right? So, I mean, I suppose you could do a chi-squared test, but it's like, the n's really small. But so it looks like they just are, it's just random, yeah. That what? Oh, I imagine these are adaptive for them to reach a frequency of 30%. I mean, right? But it's a whole chromosome duplication, not yeah. So this is that whole chromosome duplicated and reaches a frequency, that 3N version reaches a frequency of 30%, never really gets more. I mean, obviously, like, it's weird, right? Because in the sexual treatment, like, if you reach a frequency of 20%, then when you go into sporulation, there's all, that's not so great carrying 3 chromosomes through meiosis. Maybe it's fine. But so that, unlike, again, most of the yeast work where these events can happen, you're only reproducing asexually, so they're quite stable. In ARCs, if you're not a cheater, you're forced to go through meiosis every 30 cell divisions. So I guess those aneuploidies have a load associated with them. I don't know if they're dead. We don't know. No, they can't be dead, or they wouldn't be. I mean, they must be able to maintain them. No, they're 3N. So the spores would have 1 copy or 2. Yeah. Yeah, but then you'd think you could get all sorts of things. I don't know we should probably look at some of these and do genetics, but yeah, well, it's probably been done, but in other contexts. But, yeah. Mm-hmm. 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 Yeah. Yeah, this data is like 5 days old, so yeah, you're right. No, that's why it's good to talk about. Yeah, cool. Next, I don't know how long was I supposed to talk? I might be close to done anyway. Some early conclusions and work to do. These are preliminary results. Obviously, I'm trying to talk. Both my talk on tomorrow will be about pretty preliminary stuff too. The one thing that's really interesting to me is just the dominant role of rare haplotypes in this experiment. So it's like impossible to find like examples or close to impossible where we've had 2 haplotypes rip up, right? So that kind of surprised us. And that's exactly what Molly saw, but in her case, there was only, the frequencies were like 25%, 50%, or 75%. So you might say, that could just be due to sampling. But as we go with more founders, it's almost all the selections on rare variants, right? And I think, obviously, you guys have seen some rare variants ripping up too, right? Yeah. As far as I can tell, there aren't universal patterns of haplotype change with respect to time, so the data doesn't seem to really fit a model that well. So that's what we're still working on. We want to, obviously, the next experiment would be for these replicates that maintain sex to look at a more finer grained set of time points to get at what's actually happening with respect to time change. There's this observation of clonality evolving or asexuality evolving in our population. Again, I'm not sure if that's intellectually interesting or not, and I'd love to talk to people about it. The reason it might not be evolutionary interesting is that the way we do that selection for diploids is extremely artificial, and you can see a strategy that gets you through it. But Henri will probably think differently because we have had asexuality evolve. Rich Lenski could do a better job of selling it than me. Yeah. Yeah. So it is something we observe. It's extremely common, this idea of switching your sex. Again, the intervals are pretty big. We're doing some work with gene replacements and stuff like that to get at the molecular basis, but I haven't talked about that at all. Acknowledgements in my lab, the main person doing this is Rob Linder, my postdoc. He's fantastic. My technician, Polly, is obviously doing a lot of the yeast work. Several really good UCI undergraduates who I could talk at length about, who so these experiments we're doing every day. Well, so the neat thing about the experiment is that every day we're handling 384 samples. Even though it's with a robot, it's still a lot of work. We have freezers full of plates we're using. We have, luckily, the experiment's not the one we did in bacteria where we change every day. So this whole Sex on Friday thing also allows undergraduates and postdocs and faculty to have sex on the weekends as well, I assure you. The work was all, the intellectual kind of thing behind the work was a collaboration between me and Molly, right? So this is all, so she's, like, that was the first paper. If you look at the history of what happened, it was a collaboration. And Gianni Liti, who you guys have had here, is a great scientist, great lab to visit in Nice, France, was instrumental in thinking about creating these synthetic populations of yeast. One picture I like, this is that we got to make America Greta again. Okay.